Hello everyone. So last time what we talked about was the structure of a neuron, and we also talked about what a synapse is. Today we'll just look at that in slightly more detail. Let me just correct the spelling here. Right? So today we'll actually see what happens at a synapse, right? Now, previously we talked about how a synapse is the gap between two different neurons, and it is the place where the communication takes place between two neurons. Right? Now, let me show you a picture. Right, so here you have this image in front of you. Now, let me describe to you what this is. So here, what you see is that, you have one neuron here, right? This is one neuron all the way till here, really, because the axon, is all the way here. And then you have another neuron here, starting here, because these are what you see here. This is what are the dendrites, right, of this neuron. We talked about this in the previous class as well, that dendrites are basically branch-like structures that are specialized to catch any kind of signal, right, that might be coming into the neuron, right? So now you have, one neuron here. This is basically, I've circled mainly the cell body because the neuron actually goes all the way till here, all the way till here, right? Okay, now let me break this down for you a little bit more. So here you have the cell body, right? This is the cell body, right? And then here you have the axon, right? And the signal is traveling in this direction, right? Now the axon, last time we also talked about this, ends in what are called terminal buttons. So these buttons here that you see, these are terminal buttons, right? Now these terminal buttons have these little sacs, right? There are these little sacs inside the terminal buttons, which are called the vesicles, right? And these vesicles, what they carry are neurotransmitters, which are these chemical substances. We'll look at, what sort of neurotransmitters are found in the body in the next session, in the next section. But yes, so what happens is, and what you see here is that the synapse is also right here, right? Because the synapse, let me just clear this up for you a little bit. The synapse is what is connecting, right? It's a very, very small gap between the terminal buttons of this neuron, right, and the dendrites of this neuron, right? So, whatever neuron is, the neuron that is sending out a signal is called a presynaptic neuron, right? And the neuron that is receiving the signal is called the postsynaptic neuron. So here, when you see that, this neuron is what is experiencing the action potential here, this one, right? Sorry, let us just color this in red, right? So this is what is the presynaptic neuron, because here what you see is that the synapse is here, and the direction of the electrical current is in, this direction. So then this is the presynaptic neuron, and this is the postsynaptic neuron, right? And what you see here is that there is this very, very small gap between these, right? Right here, it's circled here for you as well, right? This gap is between the terminal buttons of the presynaptic neuron and the dendrites of the postsynaptic neuron, right? Now, what exactly happens at a synapse is, something that we'll see in more detail. You have a much more, detailed diagram. What you see here is that, this is the axon, right, of the presynaptic neuron, all right? And then, this is the neural impulse that's traveling, right? Then you have these synaptic vesicles, which are containing neurotransmitters. So you have these vesicles here, and what you see here is that they're carrying these chemical compounds, which are called neurotransmitters, right? Now, what happens is that, when a neural impulse travels along the axon of a neuron, right? So the cell membrane of this neuron merges with the synaptic vessels in such a way that these are released into what is called the synaptic cleft, right? So this is basically the synapse, okay? This space here is the synapse, right? And this is the cell membrane. Now, starting from here, let's do this in a different color. So this here is the cell membrane of the postsynaptic neuron, right? The neuron that is going to be receiving whatever signals are incoming, right? So this is that cell membrane, right, of the postsynaptic neuron. And here what you see is that at the cell membrane, there are these receptor sites, right? This is what you see. These are all of these receptor sites. And then what happens is that once these neurotransmitters are released, they go and they attach to these receptor sites. And they attach if they fit. So if, for example, you have these kind of neurotransmitters which are round in shape, but here the receptor site is diamond shaped. So this neurotransmitter is not going to fit in this, but it will fit into this and this, right? And perhaps, not in this shape again. But what you see here is that this is like a lock and key mechanism, right? In the sense that certain neurotransmitters will fit perfectly, whereas, other neurotransmitters may not fit as perfectly, right? So here you have this case in which the transmitter hits the receptor site and it binds to the cell membrane. And then when it binds to the cell membrane, what will happen is that an action potential, the probability of the postsynaptic neuron, right, also experiencing an action potential will increase. All right, so this is basically what happens at the synaptic cleft or the synapse, right? That you have, this axon which ends up in terminal buttons. You have all of these different, synaptic vesicles which contain the neurotransmitters. When the neural impulse travels along the axon, these neurotransmitters are released into the synaptic cleft. These neurotransmitters then go and attach themselves to receptor sites on the membrane of the postsynaptic neuron, right? And then if they attach, then probability that, the postsynaptic neuron will also experience an action potential that also increases. All right. So this is, a rough summary of what is happening, what we've just discussed. Now, what happens to the neurotransmitters afterwards? Now, that is something we can also look at. Are these 5 different stages. So, during the first stage, what you see is that, you have the storage of neurotransmitter molecules in the synaptic vesicles, right? You have an example of this here, 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 all of these different bubbles that you see here. Now, when an action potential travels along the axon here, right, then what happens is that these neurotransmitters are released into the synaptic cleft, right? So already something we talked about. Then the third step is that, these neurotransmitters bind at receptor sites on the postsynaptic membrane if obviously they fit, right? So, we talked about this earlier as well, that this is like a lock and key mechanism, right? Certain neurotransmitters will fit into certain receptor sites, just like certain keys only will open certain locks, right? Not every key can open every lock. Similarly, not every transmitter can bind at every receptor site and sort of make it more probable that an action potential will take place at the postsynaptic neuron. The fourth step is that you have the inactivation by enzymes or removal of neurotransmitters. So, this binding, it's a very, very quick process, right? And once these binds and then the neural impulse, or if there's a action potential that is produced at the, next or at the postsynaptic neuron, these neurotransmitters are released, right? And what happens is that they are inactivated, right? And they undergo this process which is called reuptake, in which what happens is that they go back to, these vesicles that they were found in earlier, right? So this is the process which is known as reuptake, right? Now we come to talking about different kinds of neurotransmitters, right? You this table, and this is also a part of your books. So the first neurotransmitter that we consider is something that we called acetylcholine. Now, acetylcholine is, this chemical substance, right? Which basically, whatever you're doing. So, for example, you're sitting, you're walking, right? You're moving your limbs, whatever you're doing, right? Acetylcholine plays, a huge part in all of these different functions that you're doing. Let's suppose you're moving your arm, you're moving your finger to write, right? So all of these motor movements that you're performing, right? Acetylcholine plays a huge part in all of these different movements, right? So basically, you have that acetylcholine is released by motor neurons, which are controlling skeletal muscles, right? So, but sometimes what happens is that even without the release of a neurotransmitter, certain chemicals or chemical compounds, right, can mimic how a neurotransmitter behaves. So let's suppose that, we don't have acetylcholine, right? But we have another chemical substance that behaves like acetylcholine in that it goes and it attaches itself. If you go back here, it goes and it attaches itself to certain receptor sites on certain neurons. And what that does is that produces an action potential in those neurons, right? So it basically behaves like that chemical behaves like acetylcholine, right? Such a chemical is known as an agonist, right? Any chemical that mimics the action of a neurotransmitter is called an agonist. Now, and one thing that we see here is that acetylcholine is not just implicated in movement, but it also contributes to the regulation of attention, arousal, right? Basically, arousal is, let's suppose, how awake you are or how aroused you are, how attentive you are, right? So when we say this, right, another thing that we see is that tobacco, right? Tobacco behaves as an agonist for acetylcholine, right? Because one of the things that you see is that tobacco can also have some sort of influence on attention and arousal, right? So basically, tobacco, we say, is an agonist, right, for ACH, because that also what it does is that chemically it mimics how acetylcholine works, right? Okay. Now, basically, so acetylcholine does all of this and it also contributes in some way to memory, right? And again, we have the same point here that, some acetylcholine receptors are stimulated by nicotine. Okay. All right. So the second the second neurotransmitter that we have, three of these, basically dopamine, norepinephrine, and serotonin, right? They basically fall within the same kind of group. And this group is what are called monoamines, right? And monoamines, what you see is that they play a huge role in the regulation, let's say, of mood. That is one of the things that you see, right? Let's look at these individually as well. So, for example, dopamine is also one neurotransmitter. And what dopamine does is that it contributes to control of voluntary movement, right? And pleasurable emotions, right? Now, one of the things that you see is that people who have Parkinson's disease, I don't know how many of you have heard about Parkinson's, but what happens during Parkinson's is that people lose control of voluntary movement, right? And that is what you see, that, dopamine, the neurotransmitter dopamine, the activity levels of of the neurotransmitter dopamine are implicated in voluntary movement. And then if there are decreased levels, then that means that is usually associated with Parkinson's disease. On the other hand, if you have overactivity at dopamine synapses, then that is associated with a mental disorder, which is an extremely serious mental disorder. 1% of the population suffers from it, and usually what happens is that in this particular mental disorder, um, functionality is so strongly affected that people end up being, sent to a mental asylum. And this is basically, this mental disorder is called schizophrenia, right? Which is marked by hallucinations, delusions. Basically, what that means is that people see things which are not there. They might hear things which are not present, right? They might have all of these different ideas which are wrong. So, for example, they might believe that they're a prophet, or that they might believe that, the president of Pakistan wants to kill them. So basically, yes. So, it's interesting to note how decreased levels are associated with Parkinson's, which is a disease that inhibits voluntary movements, right? And the overactivity at dopamine synapses is associated with schizophrenia, right? And then you have this finding that, if you have some sort of intake of cocaine, right? So that can also elevate activity at dopamine synapses, right? So certain substances also play a role in how they affect dopamine activity. All right. Then the second monoamine that you have here is what is called norepinephrine. Now let me erase all of this so that we can talk about norepinephrine. So what exactly does norepinephrine do? So norepinephrine is implicated in the modulation of mood and arousal, right? So we talked about how this particular group is, strongly implicated in mood. And again, so, cocaine and sorry, amphetamines elevate activity at norepinephrine synapses as well. What exactly are amphetamines? Amphetamines are basically these drugs that are used to, treat a number of different disorders like, you know, um, attention deficit hyperactivity disorder, or they're also in certain cases used to treat obesity. But yeah, so, basically there's some kind of drug. And they also, again, just like dopamine, can act, it can elevate activity, right, at these particular synapses. Then you have the last of these, which is serotonin, right? Now, serotonin is again implicated in mood disorders. So if you have, lower levels of serotonin or abnormal activity at serotonin synapses, that might mean that there is, that is associated with the development of depression, right? And also OCD, which is another disorder which is marked by obsessive compulsive thoughts and obsessive thoughts and compulsive behaviors, right? So yes, and serotonin is also involved in the regulation of sleep and wakefulness, right? And eating, aggression. So if you have abnormal levels of serotonin, you might not be able to sleep, or you might be sleeping too much. You might not want to eat, or you might be eating too much. You might feel extremely aggressive. So all of these are, you know, all of these are activities that are associated with what's happening with the neurotransmitter serotonin, right? Okay, so let me just make this cleaner. And then we have GABA, which is, so we talked about how there are certain chemicals that work as agonists, right? We said that agonists mimic how a neurotransmitter behaves. But we also have these chemicals that behave as antagonists, right? Which is that what antagonists do is that they're these chemical compounds, right? What they do is that they again go and they attach to these receptor sites, right? But what they do is that, in attaching to these receptor sites, they make sure that, an action potential at the postsynaptic neuron does not take place. So they basically inhibit the effect of a neurotransmitter. Let me try to explain this, that, how do they do this? So basically what they will do is that they will go and attach themselves. Let me again clean this up a little bit. What they'll do is that they'll go and attach themselves to the receptor sites that the neurotransmitter was supposed to attach itself to. So what they do is that they take the place of that particular neurotransmitter and they attach to the receptor site at the postsynaptic cell membrane, but they inhibit any sort of action potential from taking place in the postsynaptic cell membrane, right? So basically, their work is then inhibitory rather than mimicking with what we saw in the case of the agonist, right? And GABA works somewhat as an antagonist in certain cases, in the case of anxiety, right? Because what you see is that a lot of anti-anxiety drugs, right, they work at GABA synapses, right, behaving as antagonists, right? And then what you see here is that, again, it serves as a widely distributed inhibitory transmitter, right? So these are mostly inhibitory transmitters, right? Okay, now the last neurotransmitter that we talk about here is what we call endorphins. And endorphins were discovered because what happened was that you had this medical student who broke her leg, and while she was in hospital, she was being given different opiates, right, to relieve her pain. And she wondered that, if this substance that I'm being given, right, if this substance is behaving in such a way that it is decreasing pain, right? So that means that my body is responding to it, right? Which could mean that, the body itself must have some kind of natural opiate that it produces itself, right? So that, it would have these receptor sites to which, the opiate that I'm being given also goes and attaches, right? So this was her, sort of reasoning. And basically what happened then was that she set out to search for the body's natural opiate. And what she found out was that, the endorphins basically are the body's natural opiates, right? Because what they do is that they resemble opiate drugs in structure and effects, right? And they don't only contribute to pain relief, but they also contribute to pleasurable emotions. So for example, if you take a shower and you feel better, that is because there's a release of endorphins. If you go out for a walk and you feel better, it's because your body releases endorphins, which lead you to experience some pleasurable emotion, right? So yeah, so this is all for neurotransmitters and um, how basically synapses work, right?